My name is Dr. Patel, Professor and Chief Soil Health Specialist on Agriculture University and Paru University and Mati Agromat Private Limited, Alang, Gujarat. The topic of my YouTube is role of boron, molybdenum, nickel, chlorine in agriculture production. The farmers are using indiscriminate use of unbalanced fertilizer in soil for obtaining the higher production. They are not bothered for carry out the soil analysis, and they are apply the fertilizer on ad hoc basis and as per his wish or the advice of the merchant, resulted in the crop productivity has been declined at present. The farmers are apply only chemical fertilizers and they are apply only meager quantity of the organic manures. So from this table, figure 1, you can see that in 1950, we have only nitrogen deficiency, 1960 nitrogen and iron, 1970 the nitrogen, iron and zinc. So every 10 years, you can see that at present 2010 and 2020-25, we are observing nitrogen, iron, zinc, phosphorus, potassium, sulfur, molybdenum, boron, etc. And the multinutrient efficiency is increasing and the crop productivity has been declined. And the farmers are taking the intensive cropping, monsoon, winter, and summer season. And the farmers are not followed the intercropping, crop rotation. So they are taking in India, majority of the farmers are taking rice and wheat and fallow cropping skips in a year. And the farmers are unnecessarily giving the chemical load on the soil and resulted in the soil become dead, the microorganism has died due to excessive chemical fertilizer, pesticide, fungicide and herbicide. Therefore, I advise to the farmer that before application of the chemical fertilizer in soil, please carry out the initial status of soil before sowing or planting of the crop, and then accordingly, as the advice of the laboratory persons, scientists, the farmers would have to apply the nutrient, whichever nutrient is deficient or marginal in soil, to obtain higher crop production and for sustainable crop productivity. The importance of micronutrients in agriculture production. So micronutrients are required by the plant in lesser quantities, yet essential for plant growth and survival. Yield and quality of agriculture products increase with micronutrient application. Therefore, human and animal health is protected with feed of enrichment plant material. Each essential element only when can perform its role in plant nutrition properly, that other necessary elements are available in balanced ratio for plant. So we have to keep in mind that we should have to apply balanced nutrition in soil. And how we can know? Only one way, we have to carry out the soil analysis. So role of micronutrient in crop production, plant metabolism, reproductive growth, production of carbohydrate, chlorophyll synthesis, nutrient regulation, fruit and seed development. So effective function performed by the micronutrient. So any nutrient, particularly micronutrient, major or secondary nutrients, deficient in soil and yield can be declined. First, we will see the boron. So it is absorbed by plant as undissociated boric acid. Boron and molybdenum are immobile in plant. So deficiency symptoms appear in young leaves, top leaves. Boron deficiency found in sandy soil, organic soil, and fine texture bed soils. Function of boron: new cell development in meristematic tissue, pollination, fruit seed set, translocation of sugar, starches, nitrogen, phosphorus, synthesis of protein and amino acids, nodule formation in legume, regulation of carbohydrate metabolism. So lucerne crop responsive to boron fertilizers. So when your soil is deficient in boron and you apply the boric acid or borax in soil, or you spray the boric acid on lucerne crop, then the seed production of the lucerne has been increased. And we have conducted this trial and we will discuss later on. Then deficiency of the boron, since it is a immobile, deficiency symptom occurs in terminal bud growth. So you can see here, terminal bud growth, lucerne boron deficiency in lucerne. Since I told that it is a bud growth, flowering and fruit development are restricted, sterility and malformation of reproductive organ, thickened and curled leaves, discoloration, cracking and rotting of fruit, tubers or roots, apple internal cracking. Then breakdown of internal tissue in root crop given rise to darkened area referred to brown heart, black heart, cotton weaving disease. So you can see here the boron deficiency, here extreme boron deficiency in maize crops. So the crop size is also decreasing and grains are not filled properly, and there is a boron deficiency in maize crop. Then lucerne. So you can see the lucerne crop. Then response of zinc. I have conducted this trial at main forest station, Gujarat Agriculture University, Anand from 1980 to 2001, and I was serving from 1982 to 2006. So result indicate that the application of 25 kg zinc sulfate per hectare alone recorded the highest yield of lucerne variety GL1, 4961 kg per hectare, as compared to control. Application of 20 kg borax per hectare produced 461 kg, 0.2 kg lucerne seed per hectare. The percent increase in lucerne yield from 11.9 to 12.3 due to zinc and boron application respectively. The soil have marginal zinc status. So you can see here the response of zinc and boron on lucerne seed production. Then boron deficiency in cotton and boron deficiency in potato. You see here the leaf of curl, boron deficiency in cauliflower, boron deficiency in cowpea, boron deficiency in tomato. You see here the leaf are curl, here also cowpea. Then boron deficiency in mango, you can see here. Then boron deficiency in banana, dwarf plants of the banana. Then boron deficiency in citrus. Then boron toxicity in rice. You know that when the rice grown in acidic soil, then you in highly acidic soil, then you get the higher boron, and in alkaline soil also boron is more available. So there will be toxicity effect of the boron. So boron toxicity is a result of excessive boron content in soil. And the boron is more in soil, then there is toxicity. Too much boron is toxic to vegetation because plant cannot cope with the excess boron. So you will find the boron toxicity in plant. Then second element, micronutrient molybdenum. No metal anion absorbed as molybdenate. It is a weak acid and form complex polyanions such as phosphomolybdate. Plant contain less than 1 ppm molybdenum. Function of molybdenum: essential component of enzyme nitrate reductase which catalyze nitrate to nitrite. Structure component of nitrogenous enzyme involved in nitrogen fixation. So molybdenum involved in nitrogen fixation, essential for absorption and translocation of ion in plant. So nitrogen fixation not properly when there is deficiency of the molybdenum. So deficiency of the molybdenum inhibit flower formation. Imbalance various amino acid in plant. Reduce activity of symbiotic and non-symbiotic nitrogen fixation. Cauliflower whiptail. So molybdenum deficiency in cauliflower. So I told that it is a molybdenum deficiency in cauliflower was a condition called whiptail, just like tail. I have shown you so that you remember it is a tail. So whiptail in which the leaves have a thin, starpy in appearance. So you can see here leaves, normal leaves of the cauliflower. Now in deficiency of the molybdenum severe, then you can see it is just like tail one. So it is a whiptail. Then molybdenum deficiency in tomato plant, molybdenum deficiency in alpha leucine. I told you can see the patches due to deficiency of the molybdenum, leucine alpha. Then molybdenum deficiency in cannabis. Then third one is nickel. Nickel content in plant is 0.1 to 1 ppm dry matter basis taken by plant as nickel. 2. High levels of nickel may induce zinc or iron deficiency. Function: nickel metal component of urease that catalyzes reaction. Essential for nitrogen metabolism. Stimulate nodal weight and yield of crop. So nickel deficiency, hidden hunger, bougainvillea. So you can see bougainvillea, nickel can be potentially green up foliage. So hidden hunger we cannot see with naked eyes. So hidden hunger appears to be fairly common in certain ornamental species growing in artificial soil media. So when there is a hidden hunger, then we can confirm with the plant analysis, leaf plant analysis, then we can know that the deficiency of the nickel. The nickel deficiency in cannabis, it is also known as marijuana, used for drug purpose. The nickel toxicity in maize seedling. So you can see here, when there is excess nickel in soil, then you will find the toxicity. Here you can see the maize leaf. And fourth one is the chlorine. So normal concentration in plant is about 0.2 to 2% absorbed by plant as chlorine through roots and aerial parts. So it is a highly deficiency in plant because it can taken by the plant through the aerial parts. Then function, essential for biochemical reaction, osmotic cations, neutralization reaction, act as a counter ion during rapid potassium fluxes, involved in the evolution of oxygen in photosynthesis, create diseases resistant by increased osmotic pressure in cell shape, then deficiency of chlorine, partial wilting and loss of turgidity, necrosis, leaf bronzing, and reduction in growth. So chlorine deficiency in durum wheat, so it is a wheat. So you can see here the deficiency of the chlorine. Then chlorine deficiency in tomato plant. Then chlorine toxicity. So when there is a high chlorine, then scorched leaves may be the result of chlorine toxicity. Chlorine is a micronutrient essential to plant growth. However, too much chlorine can accumulate in leaf tissue. So you can see the scorched leaves in the boundary of the leaf. Tree with scorched leaves have brown or dead tissue on the tips. So it is, you can see the dead tissue on the tips margin. It is a margin or between the vein of the leaf tissue. So you can see here Leaves may be smaller than usual, so if it is normal, you can see here that they may be yellow, so it is a yellow and drop. Leaves are dropped from the plant. Then first slide I told that the farmers of the India, they are not carried out the soil analysis. So carried out soil, water and plant testing for nutrient as well as plant disease diagnosis, bacteria, fungi, nematode from soil and plant sample at present, last 1 year, I am working as a chief soil health specialist in Sevama Agri Clinic and Agri Laboratory, Bhavnagar, Gujarat, India. Our website is sevama.in. The another firm is Mati Agromart Private Limited, Bhavnagar. The website is www.matimatiagromart.com. So please contact our helpline number +91 6359595959. And we have one costly scientific instrument, ICP OES, having cost of ₹1 crores, and it is latest instrument, and we are getting the accurate results. And within last year, we have carried out more than 5,000 samples, and we have received the feedback from the farmers that the farmers are obtaining very good results after carried out the soil analysis. Because the farmers are applying the fertilizer as per soil test value and as per our advice, and the crop production has been increased starting from 15 to 30%. Then correction of micronutrient deficiency, so boron. So we are also estimated boron analysis and molybdenum. So when there is a low boron in soil, less than 0.5 ppm, then we are suggesting that we should have to apply boron powder, 20% boron powder, 5 kg per hectare. When it is medium, 0.5 to 0.7, then we suggested 1.5 kg per hectare. Then there is a sufficient, that is a 0.7 to 0.9. And if the plant show the boron deficiency, then we are recommended that apply foliar spray of 0.4% borax after neutralization with the lime on leaves on standing crop twice before and after flowering. And molybdenum, so the requirement is very low. The less than 0.05 ppm, then it is a low molybdenum category. So add 2 to 4 gram or 2 to 4 kg per hectare sodium molybdate, 39% molybdenum, or 2 to 3 kg ammonium molybdate, 52% molybdenum. The soil at the sowing planting time. Treat the seed with 10 to 20 gram of sodium molybdate or 25 kg of seeds. Medium 0.05 to 0.1. So at that time we are advising 1 to 2 kg per hectare sodium molybdate or 1 to 2 kg ammonium molybdate to the soil at sowing or planting time. And high greater than 0.1, then it is sufficient. So if you find the molybdenum deficiency in standing crop, then apply foliar spray of 0.1 to 0.2% ammonium molybdate solution 2 to 3 times at the interval of 10 days. Similarly, we are not observing the nickel deficiency and chlorine deficiency. However, I have given here. So when soil test chlorine excessive determines soil and irrigation water electrical conductivity and boron in irrigation water. So thank you very much. And if you have liked my video YouTube, then please subscribe. Thank you. If you have any doubt on soil testing, plant testing, water analysis, then please contact us. My name is Dr. P.C. Patel. My email is patelpc1212@gmail.com. Again, my name is Dr. P.C. Patel. My email is patelpc1212@gmail.com. Thank you very much again.